I will present you something we call the Tms Emg Clikr, an online platform for physiological data annotation. In order to understand what the platform is and what are the specific challenges we help to overcome, let us look at the typical Tms Emg Experimental Setup. TMS, or transcranial magnetic stimulation, is a non-invasive brain stimulation method which allows effective stimulation of the brain regions which are relatively close to the skull and scalp. In TMS, we induce a small current in the brain by creating a rapidly changing current in a wire loop, usually called the TMS coil or the TMS transducer. A changing magnetic field in turn induces an electric field, which drives charge separation inside the brain tissue and can lead to activation of neurons. However, the strength of the field decays with the second power of the distance from the coil to the target area. For this reason, we always get the strongest stimulation near the superficial regions of the brain. When performing TMS experiments, we need an easy way to detect that we have stimulated the brain. Two major requirements arise. The stimulation site in the brain should be relatively superficial, and the result should be a stimulation we can measure in some way. Probably the easiest responses to get from the brain stimulation is a muscle twitch. Activating certain parts of the brain leads to activation in specific muscles. These activations can be observed visually. However, a more quantitative way is to measure the electrical activity in the muscle using electrodes which are attached to the skin near the muscle. This is called electromyography or EMG. An electromyogram tells about the activation state of a muscle with respect to time. It is quite obvious why TMS and EMG are often combined as method. With TMS we can induce muscle activation that is readily measured by the EMG. This provides an easy framework to test hypotheses about the brain functionality and also to test different aspects of brain stimulation when we are developing new methodology. So what is this Clikr and why have we developed it? Here are a few quotes which I have extracted from various interviews with researchers. I need a reliable algorithm that detects EMG responses after brain stimulation. I want to know what is the variability between annotating muscle responses, especially the onset time of the response time after the stimulus. I see differences of even a few milliseconds in the onset time of the brain response reported as meaningful. I would like to know how accurate and reliable these measures are. I'm developing a machine learning algorithm for classifying brain responses. I need a large database of human annotated brain responses in order to do this. Our platform aims to facilitate the issues raised in these comments. And how do we do that? I will present 3 hypothetical researchers and show in which way their capabilities and needs can bind together in a synergistic way. Let us start by considering a researcher who has just conducted a TMS EMG experiment with 10 different subjects and has gathered altogether 500 potential EMG responses from each subject. There are now 5,000 responses to analyze. Some algorithms can already annotate these data automatically. However, our researcher takes their responsibility seriously and only relies on data that can provide a reliable result with a trustworthy estimate of the errors involved. Unfortunately, a proprietary algorithm provided by the software cannot guarantee these properties. Therefore, their only option is to manually go through each piece of data and annotate them by hand. Unfortunately, many of us has noted that there are not many easy-to-use tools for this purpose, and we often end up writing our own tools. With MATLAB, for example, these scripts often end up being used by a single researcher. Our tool, Clikr, provides a platform where these data can be annotated. In our preliminary test, we achieved annotation rate of up to 700 annotations per hour. With improvements and more experienced users, we expect that this number can rise even higher. In the case of our hypothetical researcher, they can upload their data to the Clikr database and annotate them using the tools we provide. I will talk about data security a bit later. Now let us turn our attention to a second hypothetical researcher. They are writing an algorithm for automated analysis of brain stimulation responses. They got a set of few thousand responses from a colleague, which they are using as a dataset. The algorithm could be a deep neural network or an autocorrelative time series analysis, or something else completely. It does not really matter in this context. However, let us assume that they want to write a deep learning algorithm that detects the onset of a muscle response. This approach relies on having numerous representative annotations available. You might recognize the problem here. An algorithm that is based on an input from a single researcher or a research lab will end up great at mimicking that researcher specifically. It might not be generalizable to the style of the other researchers. Clikr can aid in this aspect. We provide a vast dataset of annotated Tms Emg responses from a variety of researchers that can be used for algorithm development. Furthermore, our data are not annotated by a single person, but we provide multiple annotations from different researchers per each response. This vastly reduces the amount of researcher variability influencing the algorithms. Our aim in the future is to have a dataset where each response features a distribution of annotations, which can be then used to estimate the reliability of the data. This researcher greatly benefits from having this large annotated database available. Next, I will talk about how to gather these data. Our final hypothetical researcher is a group leader whose research group has been conducting Tms Emg experiments for years. Each passing graduate student and a postdoc have accumulated their own contribution to the knowledge they have on the topic. Their findings have been published in numerous scientific articles. These data, which the publications are based on, are stored on various media. Some are on laboratory servers, others are stored on memory sticks and personal computers. Altogether, there could be tens of thousands of annotated responses that formed the basis of their knowledge they gathered. What are these data good for anymore? Probably not much, to be honest. It is unlikely that they will be reanalyzed or used in another publication once the researcher that conducted the experiments has moved on. This does not have to be the case though. These data would be extremely valuable to a researcher who is analyzing brain stimulation data and also to those who are developing algorithms. The Clikr provides a way to make use of existing data that would otherwise be left unused. Data that has been published is rarely useful to keep in private. There are numerous researchers out there who are looking for this kind of data. Our hypothetical group leader realizes that there is no downside for publishing these data. The person advises the researchers to contribute all published data to the Clikr database so they become available for the community to take advantage. Now, the thing many of you will have in your mind is, this is highly sensitive biometric data, it cannot be made public. This is a sensible and good approach to data safety. We of course have put a lot of thought into this matter. We have come up with a few ground rules that should make using the database safe. First of all, we only store anonymous data. Now what this means is that when the data are uploaded to the platform, all metadata, including any dates and identifiers, are stripped and destroyed permanently. This is done on the computer of the contributor. The original data are not transmitted to the servers. At this point, we provide the contributor with an ID they can use to track each part of the data if they wish to do so. These IDs do not leave the local computer. The user can manually review the data before it's sent to the Clikr database. On the server, only raw data will be stored. Furthermore, we only store the data contributor, the accepted license, and the date of upload. Notably, we cannot retrieve any more information than that even if the contributor asks. Only possible links to the original data are left with the contributor themselves. Secondly, the data are split into standard size epochs and resampled to a fixed format. This breaks the connection of byte-to-byte comparisons between the original data and the stored data. As an additional measure, we can, for example, add a very slight amount of white noise to the data so that even comparisons between curve shapes becomes more difficult. However, we must take care not to do anything to the data that might affect the analysis results later. No one can unfortunately guarantee 100% data safety. However, let us consider two possible scenarios. If someone has full access to the Clikr databases, they can find the following information: the epoch data, the annotations given by the users, and the collections which the data belongs to. Notice that none of these data are still private. The collection states of the user who contributed the data, the license they chose, upload date, and possibly the user informations they have given. Even though having user data leak out of any system is bad, none of these are directly compromising the security of the data of any of the subjects in the original experiments. On the other hand, if someone gets access to the original data and would like to know if the subject they are looking at are represented in the Clikr database, they would first need to transform the data using the same tools as the original author, using the same keys. Then they would need to heuristically compare the epoch waveforms to each waveform in the database. While this can theoretically be done, it is computationally intensive, doesn't guarantee accuracy, and it requires access to both original data and all of the Clikr database. My aim in this presentation has been to show you that we have developed a system that provides a unique opportunity for the Tms Emg community to support each other, provide new opportunities for research, and advance the science in this field. Currently, we have a working prototype of the system running, which you can go and take a look. The link is in the last slide. We have a small example dataset running, and we are currently revising all of the front-end software. Unfortunately, the new interface was not ready for this presentation, but if you check in a few days, it should be available. I suggest that you go and take a look at the system online and consider contributing to the analysis of the database. If you'd like to contribute to the annotation of the data, you can go to the platform and start clicking. However, if you would also like to receive acknowledgement for your work, you need to create a user account to the system so we can track your progress. The user interface is still a work in progress and we'd appreciate any feedback you can provide. If you're a researcher or group leader with Tms Emg data and you would like to contribute, please contact me directly. You can find my contact information from the URL shown on the screen. Thank you for listening about the Clikr system for annotating Tms Emg data. I would like to acknowledge funding from the Janne and Aatos Erkka Foundation as well as from the European Research Council. Finally, I thank all the people who have contributed to this project in one way or another.